it is not over until it is over. Dear candidates, every minute counts. My name is Thomas MUCHIRI, and welcome to our biology lesson. During our last lesson, we looked at nutrition, and we measured in nutrition in plants. To this lesson, we are going to look at nutrition in animals. But generally, when you talk of nutrition, generally, there is when you talk of the term nutrition, just like we said, it means the process by which living organisms acquire and assimilate food. And we said that we have two modes of nutrition. We have autotrophism, we have heterotrophism. And autotrophism, this is whereby it is a mode of nutrition where living organisms manufacture their own complex food materials from simple substances. When it comes to heterotrophism, this is a mode of nutrition where living organisms, they're going to, so we have autotrophism, and we have heterotrophism. Autotrophisms, they make their own food. Heterotrophism, they feed on already manufactured food. So when you talk of autotrophism, we agreed that we have two types of autotrophism. We have photosynthesis, and as well we have chemosynthesis. Photosynthesis, this is whereby they make the food from the energy from sunlight. When you talk of chemosynthesis, they get the energy from oxidation of chemicals. Now, the other one, and as well, those organisms that exhibit this mode of nutrition, they are known as autotrophs. And we have the green plants, we have some bacteria, we have some algae. They belong to this mode of nutrition. When you come to the second one, which is heterotrophism, the one that now we are going to measure on here. Heterotrophism. As we said, that we should always introduce this or define this by saying that this is a mode of nutrition, where mode of nutrition where living organisms feed on complex food materials. And when you talk of organisms feeding on complex food materials, it means they're feeding on plant tissues and as well as animal tissues. Now, on this, which is on heterotrophism, we have 4 types of heterotrophism. So we have types of heterotrophism. We have one we have saprotrophism. We have symbiosis. We have saprotrophism. No, we have saprotrophism, symbiosis. We have parasitism. And finally, we have holozoic. So when you talk of the saprophytism, this is a type of nutrition where organisms feed on dead, decaying organic matters. Living organisms, they're feeding on dead, decaying organic matters. For the symbiosis, this is where we have the interdependence between two living organisms. They depend on each other. For example, we have the rhizobium bacteria that is found in the root nodules of the legumes. These root nodules of the legumes, they benefit. This symbiotic, bacteria, it is able to convert atmospheric nitrogen into nitrates. And it also benefits from the plant by shelter. So there is a mutual benefit. They depend on each other. We go to parasitism. Parasitism, this is where we have organisms that is feeding on another organism. So the one that is depended on is called the host, and the one that is depending on the other one, it is called the parasite. Now, we have the last one there, which is called holozoic. And this is where living organisms like human beings belong. These living organisms, they digest food. They digest food. After digesting, they absorb food. After absorbing that food that have been absorbed, it is assimilated, and then for the undigested and indigestible food materials, they are ingested. Undigested and indigestible food materials are ingested. And when you talk of undigested food materials, these are food that possibly they were not well masticated. The chewing and grinding of food was not well done. That's why possibly the service area was limited the any enzymatic digestion. Then for the indigestible food materials, these are food materials that cannot be digested. For example, the seeds of some fruits. Now, we are going to measure on this one, holozoic, because this is where we are going to get now the digestive system. We are going to go straight to the digestive system. And when you talk of the digestive system, digestion is can be mechanical, whereby we have the breakdown of food into small pieces. Then we have the enzymatic digestion, whereby enzymes, they digest or they break down food before being assimilated. Now, let's start with digestion in the mouth. So what happens in the mouth? In fact, let's start with the term ingestion. Ingestion, this is where we have the introduction of food into the mouth. Food is introduced into the mouth. So this is introduction of food into the mouth. Introduction of food into the mouth. Now, what happens in the mouth? In the mouth, we have saliva. We have saliva that helps in digestion. And this saliva, it is alkaline. Saliva is alkaline. But where do this saliva comes from? This saliva comes from salivary. Saliva comes from salivary glands. How many salivary glands do we have? We have 3 salivary glands, and these salivary glands occurs in pairs. The first one, we have the parotid. We have the parotid gland. The second one, we have the sublingual. We have the sublingual gland, and lastly we have submandibular. We have submandibular. And for the first one, the parotid, it is found in front of the ears. Then the sublingual, it is found beneath the tongue. Then submandibular, it is found under the jaws. Now, this saliva contains what? What are the components of the saliva? Components of saliva. Saliva is composed of one, what we call the enzyme, the salivary amylase, and this salivary amylase digest starch to maltose. Digest starch into maltose. Something else, we also have mucus, And the mucus has got two roles. The first one, it is lubricating food. So we have it lubricate food and mouth lining. Lubricate food and mouth lining. And finally, it also helps in binding these food particles together. So it also helps in the formation of the boluses. So binding helps in binding the food particles together. Mouth, we still have teeth. These teeth help in chewing, and grading of food. This chewing and grading of food is known as mastication. It is known as mastication. Grading of food is known as mastication. What is it that is remaining in the mouth? We have the tongue. What is the role of the tongue in digestion? The role of the tongue in digestion, we say that it helps in formation of the boluses, and also pushing these boluses to the back of the mouth. So this one, it also pushes the boluses to the back of the mouth. So these are the two functions of the tongue: formation of the boluses and also pushing these boluses to the back of the mouth. Now from there, we are going to, after this food has been pushed to the back of the mouth, there's a soft palate. Soft palate raises up, and then the food passes. After passing the soft palate, there is a structure known as epiglottis, and this epiglottis, it helps in directing of the food into the gullet. So it directs the food into the gullet. To direct the food into the gullet. Now, and when this food gets into the gullet, we find that now gullet is as well adapted. So now when you talk of the gullet has got muscles. These muscles, we call them, has got longitudinal and circular muscles. Longitudinal and circular muscles. These two muscles, they are the ones that initiate the wave of contraction. These two muscles initiates the wave of contraction. It initiates the wave of contraction. And the wave of contraction is the one that we call peristalsis. This wave of contraction is the one that we call peristalsis. So this is all about this is what happens in the mouth. So starting from there, we have said that the food is introduced into the mouth. After food being introduced into the mouth, there is in the mouth we have the saliva, and we have said saliva is alkaline, and this one, it creates the best pH for the digestion of starch into maltose by the salivary amylase, or what we call ptyalin. So we have the three salivary glands there. We have the parotid gland, we have the sublingual gland, And we have said that parotid gland, it is found in front of the eye or in front of the ears. We have the sublingual, it is found beneath the tongue. We also have the submandibular that is found under the jaws. Then we also have the components of the saliva, which is the salivary amylase that digest starch into maltose. We also have a mucus that replicate food and the mouth lining, and as well it also helps in binding the food particles together. Now, water. We have said that this one is a solvent. This is where the food substances dissolve into before being absorbed into the body. Water as well, it softens the food. And this softening of food, it also helps in mastication when it comes to chewing and grinding of food. Now the tongue, the two functions. One, we have the formation of boluses and also pushing these boluses to the back of the mouth. And after the pushing by the tongue of the bolus, after pushing these boluses to the back of the mouth, the soft palate raises up, and now food passes the nasal cavity, and now it gets, there's a structure there called epiglottis that directs the food into the gullet, not to get into the trachea. So now from there, we have said now the gullet has got longitudinal and circular muscles, and these muscles, they're the ones that initiates the wave of contraction, and this wave of contraction is the one that you call peristalsis, whereby now the food can be now swallowed. Now, before this food gets into the stomach, there's a muscle that controls whatever is getting into the stomach, and we call it the cardiac sphincter muscle. We have the cardiac sphincter muscle. This muscle, it controls whatever is getting into the stomach. It controls whatever is getting into the stomach, and also the content of the stomach, not to move up into the gullet. So now, after this food gets into the stomach, its presence, it stimulates the production of gastrin hormone. Presence of food. So the presence of food stimulates. That's the best term. It stimulates production. It stimulates the production. It stimulates the production. Stimulates the production of gastrin hormone. Stimulates the production of this gastrin hormone. And what is the role of this gastrin hormone? We understand that this process, starting from the mouth, for you to salivate, for you to get this saliva, the presence of food is going to stimulate your body to produce this component, the saliva. The presence of food into the stomach, it is going to stimulate the production of gastrin hormone. What is the role of this gastrin hormone? Gastrin hormone, it helps, or it is the one that stimulates the production of gastric juice. So it stimulates the production of the gastric juice. And now that gastric juice is the one that usually have the four components. We have the HCl, renin, pepsin, and as well mucus. So let's see what is going to happen in the stomach. What is going to happen in the stomach? So now we have said that we have the gastrin hormone. So gastrin hormone stimulates the production of gastric juice. Stimulate the production of the gastric juice. Now, this gastric juice, it is produced by the gastric glands in the walls of the stomach. What are the components of the gastric juice? So we look at the components of gastric juice, and we are as well going to look at their function. What is the role of these components of the gastric juice? So we have the first component there. We have two enzymes. The first one is we have renin, we have pepsin, and these two enzymes, they are produced in precursor form, or they are produced in inactive form. So they are produced as this one is produced as prorenin, and this one is produced as pepsinogen. It is produced as pepsinogen. We also have three other components. We have the mucus, and we also have the hydrochloric acid, which is the HCl. So we are going to look at the function of each component. So for renin, this one mostly is found in young babies who are suckling, because it digests milk proteins. So digest milk proteins, the one that we call carcinogen to casein. These are the milk proteins, from carcinogen to casein. Pepsin digests proteins. This one digests proteins into peptides. Digest proteins into peptides. Then we also have the mucus. This mucus is the one that now forms a thick protective lining on the walls of the stomach. Thick protective lining on the walls of the stomach. And this lining, it is the one that now prevents corrosion. Prevents corrosion of the stomach walls. The stomach walls by the HCl. It's the one that prevents the corrosion of the stomach wall by the HCl. It as well helps in lubricating the food. It also helps to lubricate food, helps in lubricating of food. Now, the last one, we have now the HCl. HCl has got three functions. One is that it is the one that now activates these prorenin to renin, conversion or activation of pepsinogen to pepsin. So one of the function of HCl, we say that activation of enzymes. We have activation of enzymes. Then, other than activation of enzymes, we also have this HCl also helps to kill bacteria. Then it also creates an acidic medium for the action of enzymes. For these enzymes to function, they require an acidic medium. So creates an acidic medium. It creates an acidic medium for action of enzymes. action of enzymes. So basically, this is what happens in the stomach. Digestion of milk proteins, carcinogen to casein, digestion of proteins, that is proteins to peptides, and you also have the mucus there that also helps in corrosion, prevents the corrosion of the stomach walls by the HCl. It as well helps to lubricate the food. Now, HCl activation of these digestive enzymes, pepsinogen to pepsin, prorenin to renin, create an acidic medium for the action of enzymes and as well killing of the bacteria. What else happens in the stomach? In the stomach, we find that this stomach also have got muscles, and these muscles, they also have a role to play in the process of digestion. So the stomach have the two muscles there have longitudinal muscles and circular muscles. And these longitudinal muscles, longitudinal muscles and circular muscles, they help in mixing of food. So help in mixing of food. And this mixing of food, we call it churning. This is what you call churning, the mixing of food. And now, when that food now has been mixed, it forms, this one now forms what we call chyme. So chyme is the end product of mixing this food or churning this food. Now, from there, this food now, it is held by a muscle on the lower side, so that now it do not move into the duodenum. And this muscle that we call it a pyloric sphincter muscle. It is the one that controls whatever leaves the stomach. It is the one that controls whatever is going to leave the stomach. So whatever is getting into the duodenum. Now we go to the digestion in the duodenum. We have now duodenum. What happens now in the duodenum? Just like we started in the mouth, presence of food stimulating salivation, and the presence of food in the stomach stimulating the production of the gastrin hormone, the one that is going to stimulate the production of the gastric juice, and the gastric juice has got these components. So presence of food in the duodenum, it is going to stimulate the production of two hormones. So we say that presence of food stimulates the production of two hormones, and these are the two hormones. The first one is secreting hormone, and you also have cholecystokinin hormone. Cholecystokinin hormone. So these two hormones, the presence of food, it is going to stimulate the production of these two hormones. And the two hormones, they are produced by the walls of the duodenum. These two produced by the walls of the duodenum. And these ones, they are produced, after being produced, they are introduced into the bloodstream. Now, what are the roles of these two hormones? So the first one, secreting hormone, the role of secreting hormone. So secreting hormone is going to stimulate the secretion is going to stimulate secretion. Is going to stimulate the secretion of the bile juice from the gallbladder, into the duodenum, through the bile duct. What does this one means? This one means that now in the liver is the one that produces the bile juice. So in the liver, so if this is a liver, so bile juice is produced by the special cells of the liver, and it is stored in a structure that we call the gallbladder. Now this gallbladder, so it is the one that is storing the bile juice. So it contains the bile juice, but this bile juice is required in the duodenum. So we are saying that secreting hormone, it is the one that now stimulates the secretion of the bile juice from the gallbladder into the duodenum. So this one is a duodenum through the bile duct. So this is a function of secreting hormone. So this bile juice is required in the duodenum for the now let's look at the functions of bile juice. The bile juice. So one, the bile juice is going to break down. The large fat droplets into tiny fat droplets, what we call emulsification. The first one is emulsification. And the importance of emulsification, it is to increase the surface area for the enzymatic digestion of the lipids. Now from there, it also helps to neutralize the acidic chyme. Whatever is coming from the stomach, because of the HCl, it is acidic. So it helps in neutralizing the acidic chyme from the stomach. So it neutralize the acidic chyme from the stomach. Acidic chyme from the stomach. Now, what is this that this bile juice contain, so that now it can help in carrying out these two functions? So the components of the bile juice, there are two. We have, so we're going to look at the components of the bile juice. Components of the bile juice. There are two salts. That's why They are neutralizing the acidic chain because they are alkaline. So components of the bile juice. So there are two. The first one, we have sodium taurocholate. We have sodium taurocholate. The second one, we have sodium glycocholate. So these are the two salts that carry out this emulsification and as well as neutralizing the acidic chain. So now those are the two the function of secreting hormone. The function of this secreting hormone. Now we have cholecystokinin, the other hormone, cholecystokinin. So what are the roles? The roles of cholecystokinin. The role of cholecystokinin, it is the one that stimulates the production of pancreatic juice. So this one, it stimulates the production of the pancreatic juice. So it is this cholecystokinin that we get in the pancreatic juice. Now, what are the roles? What are the components? We start by components. Components of pancreatic juice. Components of the pancreatic juice. So the first one, we have the pancreatic lipase. We have the first one is pancreatic lipase. We have pancreatic lipase. Number two, we have pancreatic amylase. Then we have trypsin, and number four, we have sodium hydrogen carbonate. We have the sodium hydrogen carbonates. So for the production of this pancreatic juice, it is under the influence of cholecystokinin. So maybe we started here, the duodenum. Presence of food stimulated the production of hormones. These two hormones, we have the cholecystokinin and the secreting hormone. What is the role of these hormones? The secreting hormone, it helps in to stimulate the production of the bile, secretion of the bile from the gallbladder into the duodenum through the bile duct, as we can see here. Then we have the other one that we are saying that the components of the bile juice, we have the emulsification, which is a breaking down of the large fat droplets into small fat droplets. So that is what we call emulsification. Then lastly, we have neutralizing of the acidic chain, that is in the stomach. From there, we look at the components of the bile juice, which we have the sodium taurocholate, sodium glycocholate. These two, they are mineral salts. The roles of the other hormone, cholecystokinin, production of the pancreatic juice. Then we have the components of the pancreatic juice. we have pancreatic lipase, we have pancreatic amylase, then we have trypsin, and we have sodium hydrogen carbonate. So we are going to look at the roles played by these hormones, by these juices. So now we are going to look at the roles. We are going to look at the roles. We are going to look at the roles of the components of the pancreatic juice. So now these components, the first one being pancreatic amylase. What is the function of the pancreatic amylase? Pancreatic amylase, it speeds up the breakdown of the remaining starch, remaining starch into maltose. The breakdown of the remaining starch into maltose. Remember that the breakdown of starch, it started in the mouth, whereby salivary amylase broke undigested starch into maltose. Now the other one is pancreatic lipase. Pancreatic lipase. This pancreatic lipase, it digests. This one digests lipids into fatty acids and glycerol. It is good to note that one molecule of lipid is made up of 3 fatty acids and 1 glycerol. The other one is trypsin, and it is good to note that trypsin is produced as well in precursor form, which is, so we have, it is produced in form of trypsinogen, and then it is activated to pepsin. What activates this? It is called enterokinase. Enterokinase is a cofactor. Among the factors that affect enzymatic digestion, we have the cofactors and the coenzymes. These are cofactor. Cofactor, it is meant to activate the enzyme. So we have trypsin digesting. So trypsin digests proteins into peptides. Digest proteins into peptides. Then we have sodium hydrogen carbonate. We have sodium hydrogen carbonate. And this sodium hydrogen carbonate, it helps to neutralize the acidic chain. So it also helps to neutralize the acidic chain. From the stomach. So those are the functions of the components of the pancreatic juice. Now, from here, the food now moves into this small intestine. Small intestine. What happens here? In the mouth, presence of food, Seeing the food, it stimulates salivation. Stomach, presence of food in the stomach, stimulate the production of gastrin hormone. Duodenum, the presence of food stimulated the production of cholecystokinin and secreting hormone. As well, in the small intestine, the presence of food in the small intestines stimulate. So presence of food stimulates. It stimulates the production of intestinal juice. The intestinal juice is known as suckers entericus. It is known as suckers entericus. Suckers entericus. Now, this suckers entericus, this whatever you're calling the intestinal juice, has got the suckers entericus has got 5 components. So what are the components of these suckers entericus? So let's see. So we have suckers entericus components of suckers entericus. We have the first one. We have maltase. We have lactase. We have sucrase. We have lipase, and we also have peptidase. So the function of these enzymes, because they are enzymes, the first one is breaking down the food, which is maltose to glucose. Then this one, lactase, digest lactose to glucose and galactose. The other one, sucrase, digest sucrose into glucose and fructose. Lipase, digest lipids to the fatty acids and glycerol. Then we have peptidase that digest proteins, that digest peptides. These digest peptides into amino acids. This one digests into amino acids. So these are the ones that are absorbed into the body. So basically, this is where the end of digestion. These are where the digestion stops, because these are the components that are digested, absorbed into our bodies. And from there, the last part is duodenum, is the large intestine, where we have the absorption of water, and you also have the bacteria that helps in the synthesis of vitamins like vitamin B1, B2, B12. Those ones, they are produced under the influence of the vitamins. They are produced in the large intestines. So this marks the end of our lesson, that is digestion. There is maybe much that you need as well to look at. For example, the adaptations of each and every part, starting with the mouth, the small intestine, the stomach, large intestines, all those. So I would like to take this opportunity to thank you, the viewers, and for the candidates, we wish you all the very best. Have a good time.